Welcome back, friends. In this video we are going to discuss about Hardy-Weinberg Principle. So what will be Hardy-Weinberg Principle? So this is very important in the case of CBSE board exam as well as in the case of NEET examination. NEET point of view also will be getting problematic questions from this topic. So what is the concept actually here means that the idea is about, suppose you can imagine a population of 5,000 people. So in the population of 5,000 people, how many times the allele is repeating, okay? The allele frequencies as well as the genotypic frequencies. So based on this, they will be calculating, they will be giving some equation. That means without evolutionary consequences, always this Hardy-Weinberg principle will be constant, that will be equal to 1, that will not at all change. So easily how you can remember, for example, you can take chromosome, okay? So this is the first chromosome, for example. So in first chromosome you have some allele is present here. For example, you can take allele T. For example, you can take allele T is present in this location in chromosome number 1, okay? So this is for one person. So the population is about 5,000 people. So randomly you are choosing another person. So the second person is having, again you can see here the second person. The second person is also having T. Okay, you can see here he also having the T, allele T, at the same location of the same chromosome. That is the second chromosome. Okay. So again randomly you are going for some other person. So in the person also, for example, you can see here this is for example third person. Here he is having the allele, but that is not T, that is small t. The allele is different in the third person. Okay, this third person, but all in the same chromosome. First chromosome of the person. Actually, we all know we have 46 chromosomes. That is 23 pairs of that. So you can see in the group of population, two persons are having the same alleles at the same location, but the third person is having the different allele at the same location. So this is allele frequency you have to calculate. So allele frequency will be, so this will be more and this will be less. So in a population, how many times the allele is repeating in the same location of the same chromosome, that is called as allele frequency. What do you mean by allele? Allele is an alternative form of gene. Okay, suppose for example you can take height. Okay, height is a single character, right? Height means that is a denotes the height, that is single character. For the single character you will be having alternative forms. One may be very tall, one may be very short. So two alternative forms are there. So you can mention like capital T and small t. So two alternative forms will be there. These alternative forms are called as alleles, but both will be for height, the single character. This together is called as genotype. Okay, so this is called as genotype. When you individually take T or P, that is called as allele. So this is allele, and together if you take, that is called as genotype. For a single gene you will be having two forms, alternative forms. Suppose if you take a film, movie. So movie as a gene, in a movie you will be having hero as well as villain. So alternative forms will be there, right? Similarly here, gene is a hero. So alternative forms will be there. One will be dominant, one will be recessive. In movie also the hero will be dominant, the villain will be recessive, right? So the same thing will be here also. So this is what the category. So in Hardy-Weinberg principle, they are going to calculate genotypic frequencies as well as allelic frequencies. So if you take the population, the gene pool, right? So you'll be calling it as gene pool. Gene pool means total number of genes in the population, that is total number of genes as well as alleles in the population remains constant, that will not at all change, okay, until it is disturbed by any of the like evolutionary consequences like mutation, natural selection, recombination, genetic drift. Many are there, I will be discussing in the next class. So many consequences are there. If evolutionary consequences are not involving there, the population will, the genes and the alleles will be constant for generation after generation also. For example, in the 5,000 people, so imagine 2,000 people are having TT. Okay, so this is like genotype, for example. So remember 2000 are having TT. So 1000 people are having capital T small t, like this will be having. This is homozygous condition. This is also homozygous condition. Homozygous means so two alleles will be the same. So here two alleles are different. So this is heterozygous condition. So they will be having, this is the combination. So this is the frequency of genotype in the population. So this is the percentage. Okay. So when you go to the next generation, the population level will increase, but the percentage of the genotypes will not change. Okay, that will be the same. For example, after a few generations, 1000. Okay, 10,000 people are there. So 10,000 people are there, right? So that again, if you go for the percentage of the genotypes, so TT will be having, for example, I will be writing here. So 4,000 will be TT. 4,000 will be small t and small t, and 1,000 will be small t and capital T, like this. So again you can see here the frequency, the percentage is similar. So whenever the generation after generation, the frequency of the genotype repeating in the population, I said no, in the particular chromosome, particular location, the allele as well as genotype, that is repeating how many times, that is called as frequency of alleles or frequency of genotypes. Alleles means only single we will be taking. Genotype means, so we can write like this, TT. So TT, again TT. So this is genotype. So this is allele frequency, genotypic frequency. Here you have to calculate like this, right? So for this they have given a formula. So imagine they will be giving like P. So P denotes the frequency of the dominant allele. So P means frequency of the dominant allele. Which is the dominant allele here? Capital T is the dominant allele. So Q denotes the frequency of the recessive allele. Which is the recessive allele here? This small t is the recessive allele. So recessive allele. This is the frequency of the dominant allele. This is the frequency of the recessive allele. So what I said? So always it remains constant. So we can write it as P plus Q is equal to 1. So this is the equation here. This is for allelic frequency. So allelic frequency will be like this. So what will be the cap P? That will be the dominant allele. This will be the recessive allele. P plus that is equal to 1. So in a population you can take, for example, an allele. So here in a population of 10 members, okay. So 8 members have capital T, 2 members have small t. So how you can write the equation here? So when you convert into percentage, they will be having 0.8 plus 0.2 equal to 1. That means 1 equal to 1, that will be balanced. That is equal. RHS, right hand side is equal to left hand side. So that will be equal. It will be disturbed. Suppose there is any like evolutionary consequences, that will change. So this is equation for allelic frequency. When you go into the genotypic frequency, I will be writing here. When you go into the genotypic frequency, so we can write like this, right? So this is for single allele. When you go for genotype, you will have alternative forms of alleles. So you can write it as P plus Q the whole square equal to 1. Why P plus Q the whole square? So this is one allele, right? So you have two alleles. So P plus Q equal to 1, right? So this is for one allele, this is for another allele, the combination. So P plus Q the whole square. Now we can apply the formula A plus B the whole square, right? That formula we can apply. So when you are applying the formula, you will be getting A square plus B square plus 2AB, right? So you will be getting what? P square plus Q square plus 2PQ. That is A square plus B square plus 2AB is equal to 1. So this is the equation you are getting for this genotypic frequency, right? So this equation should be there. So now we can go for the genotypic frequencies. Okay, P square. What is P square? What is the dominant? P denotes the dominant allelic frequency, right? So P denotes what? Dominant allele T square. Q means what? Recessive allele. So T square, and plus 2 into P means capital T, Q means small t, equal to 1. So when you simplify this, TT plus 2TT equal to 1. So this is a simplified form of the equation. So always it remains constant, that will never change. When you go and apply into the Mendel's equation now, so you'll be getting the answer clearly. So I'll be writing here Okay, it is very easy. So when you go for Mendel's equation, you will be getting right like this, right? So this will be T and T. Here also will be having T and T. So Mendel's monohybrid cross is the right. In that you will be getting like this. So and T and it will be the monohybrid cross. The checkers board is this Punnett square, right? So this is the dominant allele. This is the dominant allele, and this is the recessive alleles. So when you calculate the genotypic ratio, you will be getting 3:1, right? Phenotypic will be 3:1. Genotypic will be 1:2:1 will be getting. So now, as we already calculated the frequency, that is 0.8 for capital T, 0.2 for small t. Previously I said, right? So in the P plus Q equation I have given equal to 1. So 0.8 plus 0.2 equal to 1. So this is what we got already. Now we have to substitute it here. So 0.8 here in this location, here also 0.8, here also 0.2, here 0.2. Here what will be coming? P and T. So 8 will be 0.64. T and T. So 0.8 and 0.2 will be 0.16. And here you'll be getting T and T, that will be 0.16. Again here and here, small t will be 0.4. So this is what you got here. Now when you substitute this equation here, you can get the answer. TT. That means what? TT value will be 0.64. Small t value 0.4 plus 2 into TT. Okay, this TT is there, right? TT value is what? 0.16. That is a TT value equal to 1. When you simplify further, 0.64 plus 0.4 plus 0.32 equal to 1. When you simplify, you will be getting 1 equal to 1. So this is how you have to calculate. So always this will remain constant until the evolutionary sequences are not playing in that case. So how means, for example, I said already, right? 80 people or 80% is having T and 20% is having small t in a population. Suppose there is any evolutionary consequences like mutation occurred. As a result of mutation, this may increase to 10%, so that becomes 90%. This becomes only 10%. Okay, so there will be the alteration, there will be change. So or else from here to another population they may migrate. So many consequences are there, like 5 factors are there. Regarding these 5 factors, I will be explaining you in the next class. So this is about the Hardy-Weinberg Equilibrium. Very simple. Only 2 equations you have to keep in mind. This is for allelic frequency and this is for genotypic frequency. So by this way you will be getting many problems. Simply you can substitute here and you get the answer. Thank you friends. Thank you for watching this video.